Okay, here we go. About alimentary tract, please be very clear about the difference, primary and secondary. You're going to get questions about those, right? I'm not going to answer, give you an exact things that shouldn't be written here, but know what is the difference. Peritoneum, understand the difference between visceral and parietal, right? I'm not going to ask you which organs are retroperitoneal and which are not, but you have to understand what are retroperitoneal, the ones that are posterior to the peritoneal cavity. Am I clear? Role for mesentery, supply, blood supply and lymphatic vessels and nerves. That's all you got to know. Now, digestive activities. Remember we had the question something like, what are the digestive activities of that you can call, what is the word? Propulsion, mechanical, okay, mechanical digestion, okay? Or what is propulsion? Peristalsis and deglutition, for instance. Okay, so just be able to group them together, okay? Propulsion, yeah, so it's going to be deglutition and peristalsis. What is necessary for chemical digestion? Secretion of juices, okay? Mechanical breakdown, we're talking about churning and segmentation. Now, for external stimuli versus internal stimuli, basically, you have to be able to recognize those. External stimuli will be what? Something like a smell or a taste, right? And internal will be either mechanical stimuli, distension, correct? Or chemical stimuli like food chemicals or decreased or increased pH. We're clear about it. I also want to be very clear about this. So, digestive activities. External stimuli. What do they act upon? Which part of the nervous system? Huh? Central nervous system. Now, internal stimuli act on peripheral autonomic nervous system, right? Autonomic. You have to understand that actually all digestive reflexes, all of them, are they autonomic or somatic? Huh? All digestive reflexes, autonomic, no exceptions. Okay. Now, the sensory input, yes. So Joseph correctly noted. Sensory inputs from external stimuli will end up in the CNS, in the brain. Sensory inputs from internal stimuli will mostly, vast majority of them, will end up where? Do you know what is the pH of your stomach right now? Do you know? I don't. Okay? So it's going to be medulla, spinal cord. Local plexuses. Does that make sense? Now, motor outputs. What are the effectors? All this complicated wording refers to what are the effectors of the autonomic reflexes, of the digestive reflexes. What are the two effectors of nervous system? In general, muscles and glands. So glands, obviously, secretion of juices, and what else? Which muscles? Smooth. That's it. Cool. And yeah, we're gonna review the quiz at some point. Yeah, no worries. Internal stimuli, basically understand, distension and chemicals, food. Okay. So what types of receptors? We're talking mechanical, chemical, osmoreceptors. So you have to understand that you have a distension, that acts on mechanical receptors, and food chemicals will act on chemical receptors, not the other way around. Sensory inputs, which just discussed. Okay. What is the gut brain? That's a nerve plexus in the walls of the stomach or in the walls of the intestine. Okay. What are the 4 layers commonly found? Okay. From deep to superficial. From deep to superficial. Mucosa, submucosa, muscularis externa, serosa. Mucosa consists of, what we're going to do, we're just going to review this whole thing, okay? And pretty much skip through many of these questions. So, mucosa consists of epithelium, okay, what else? Lamina propria and muscularis mucosae. Then you have submucosa, which is basically connective tissue with a lot of blood vessels. Then you have muscularis externa. How many layers usually? 2 of what? Smooth muscles, circular and longitudinal. And then you have serosa, which produces watery secretion to avoid extensive rubbing and friction on the outside of the organs. Okay? This is going to be, pretty basic. Right? So please know those substructures. Please. Like, I ask you, where you're going to find smooth muscles. You tell me it's going to be either muscularis externa or muscularis mucosae. We're going to find lamina propria. That's going to be mucosal layer. In the mouth, what happens? Mastication mostly, right? Also, mouth participates in deglutition and it pushes the whole thing to the pharynx. Anatomical cavities. Now, this is the anatomy portion. Let me spare you from anatomy portion, like oral cavity proper, from buccal or vestibular cavity. You all can know that. I'm not going to talk about anatomical structures, really. I also promised, and I will keep my promise, pictures that you're going to see on anatomy exam. You saw them. They're in the PowerPoint. Okay? 2 parts of Palatine, soft, hard. What's the function of uvula? Closing the nasal cavity, nasopharynx, when you swallow. Structures of the tongue responsible for the taste. It's going to be papillae, taste buds on the papillae. 3 types we have talked about: fungiform papillae, valvulate papillae, and foliate papillae. What are the functions of palatine tonsils? They protect immune function. Differences between intrinsic and extrinsic salivary glands. Intrinsic are single-celled, extrinsic many cells. Intrinsic salivary glands located in the lips, cheeks, right in the tongue. Enzymes do they release? What do they release? What 2 basically enzymes that you're going to find in the saliva? Lipase and amylase. Lipase breaks down fat. Amylase breaks down carbs. 3 extrinsic salivary glands. Yes, they can be on the anatomy exam. Parotid, sublingual, submandibular. Parotid is the largest. Okay. 2 types of secretions. Okay, yeah, that's okay. 2 types of secretions that saliva is made of: mucus and serous, right? Mucus is mucus, and serous is watery. Mucin, sticky stuff. Mucin is sticky. Water, what does it do? Dissolves food chemicals. Lysozyme. Antibacterial immunoglobulins. Antibacterial, antiviral, lingual lipase, salivary amylase. Amylase just told us. Breakdown of fats and carbs. Okay? So, which branch of nervous system activates salivary glands? Sympathetic or parasympathetic? Parasympathetic. Fairly easy. You can remember this way. Rest and digest is the function of parasympathetic system. So if any part of digestive system is activated, that's due to parasympathetic. So sympathetic will inhibit. Can you describe the salivation reflex? Okay, go. What's the stimulus? Anything, huh? Taste, presence of food in the mouth. What happens next? Stimulates salivation center in medulla. Medulla produces the motor response. You salivate. That's the depth. You have to know it. Okay? When you're excited, when you're excited or frightened, what happens to your saliva? It becomes thicker. Why? Vasoconstriction of blood vessels that run, because where do we get water for saliva? From the blood. If salivary glands are removed, what you're not going to have? Specific functions. It will be hard to swallow because food is not sticky. It wouldn't be moist. Arguably one of the most disgusting words in English language, the word moist. So it would be hard to swallow. You would lose the digestive activities in the mouth, right? And your teeth will get totally destroyed because you would not have antibacterial activities. Okay? Peritonitis is inflammation of salivary glands, extrinsic salivary glands, caused by virus. In males, it can cause infertility because of complication on testes. And prevention, there's no treatment. You can vaccinate. Tooth, top to bottom. Crown, neck, and root. Layers. Enamel, dentin, and then pulp. So in the pulp is in the pulp cavity, and what you're going to find in the pulp? Nerves and blood vessels, right? Okay, sorry. Types of teeth. Incisors or incisors. Canines. Premolars and molars. Which ones are absent in deciduous teeth? Premolars. Okay. When does pulpitis happen? When bacteria get into where? Pulp cavity. Yeah, right. Cavity. Gums are connective tissue. Okay. By the way, gums are covered with what? Epithelium. Stratified squamous because rubbing, right? When I say where's pharynx anatomically, it's really hard to describe. Let me ask you this. Do you understand where it is? The pharynx? Do you understand 3 portions of the pharynx? We'll be able to identify them on the anatomy exam. Good. Anatomy exam, by the way, as usual, multiple choice. What are the 3 parts? Naso, oral, laryngo. Which ones participate in digestion? Oral and laryngo. So they covered with what type of epithelium? Stratified squamous. And naso is pseudostratified. Okay? Pseudostratified columnar. Pseudostratified columnar. How many deciduous teeth, by the way? No, deciduous. 20. Permanent 32. Pharynx. Structures that bound the pharynx. What I mean by that? How do you get there? From where? Food, how does it get in the pharynx? From the mouth, and then, if you're lucky, it goes where? Esophagus. If you're unlucky, it goes to where? Larynx and trachea. If you're unlucky, like really unlucky. Okay. Yeah. So this, I mean, you kind of have to appreciate that, you know, since food comes through the oral and laryngopharynx, it's stratified squamous epithelium. Now, let me swallowing reflex. So what happens? Tell me. First, when you swallow, what happens? No, first. What happens first? Huh? You push food back into the, which part of pharynx? Oropharynx. What happens next? You feel the kind of it's mechanical stimulation. So that stimulation activates what? Deglutition center in the medulla. What does medulla do? Closes the by uval it closes nasopharynx, and by the means of epiglottis it closes the larynx. Opens esophageal sphincter, and the food gets into the esophagus. Know the sequence. By my behavior, you can tell. I will ask something about the sequence on the exam. Okay? Am I clear? So esophagus joins stomach at, anyone remembers, orifice? Cardiac orifice. And it's separated from stomach by cardiac sphincter, gastroesophageal sphincter, lower esophageal sphincter. There are many names. So basically that sphincter controls the motion. So when you jump with the full stomach, the food doesn't pop out of your stomach back in the esophagus. Esophagus, how does it look when there's no food in it? It's collapsed. What are the layers? What is the difference between esophagus and like stomach? Couple differences. So first of all, does esophagus have serosa? No, it's adventitia, it just attaches it to other structures. What else? The M word. Muscles. The upper third is skeletal. The middle third is mixed. The lower third is smooth. We good? And of course, what type of epithelium you can find in the esophagus? Stratified squamous, of course. We got it. We got it. We got it. Does esophagus have sort of? No. What happens to the epithelial? Okay, when esophagus is over and stomach starts. At this junction, what you can see? Stratified squamous on the esophagus side and just abruptly, what type of epithelium covers stomach? Simple columnar. Basically, you can say, look, it's fairly easy to, from the moment it goes here to the cardiac orifice, it's stratified squamous. At cardiac orifice, it changes to simple columnar and goes like this to the anus. And then it changes to stratified squamous again. That's it. Okay? You need to remember these two turning points. Parts of the stomach. Cardia, that's upper. Fundus and butt. I would put fundus and body together because they functionally similar. Is that okay that I'm sitting? Because it's really impossible to draw all the time. So fundus and body are similar. And then pylorus, which can be separated into the antrum and pyloric canal, which leads semi-digested food to the separated pyloric sphincter, which controls the passage of food. We good? Rugae just folds, basically, okay? Layers. Okay. What is the only difference? What stomach has that nothing else has? No. I'm talking about layers of the stomach. They have like extra layer. Third layer of smooth muscle, oblique. Okay. Now, in terms of the stomach, identify parts. Am I clear? Well, Rugae is kind of dumb. I mean, come on. And no, I will not put the arrow somewhere in the ballpark of pylorus and will ask you, am I pointing at the antrum or canal? No. But, I mean, you should be able to tell apart, say, antrum from fundus, pyloric antrum from fundus, or, you know, cardia from pyloric canal. Does that make sense? Yeah. Difference between, third layer, epithelium, simple columnar, types of cells. Okay, this is important. So, which cells? Mucus cells. So you need mucus. What else? You need, stem cells will be found to make new cells since old ones die all the time. What else? Parietal cells, which produce acid. Chief cells, which produce pepsinogen. Very good. That's a good answer. Not pepsin. They produce pepsinogen, which is converted into pepsin in the stomach lumen. Enteroendocrine cells, what do they produce? H word. Hormones. Which, well, we'll get to hormones. We good? Specifically, G cells, what do they produce? Gastrin. Okay. Now, in the cardia, what type of secretion would you expect? Mostly. I mean, what is produced? You have mucus, acid, pepsinogen. What will be produced in cardia mostly? Gastrin, you know. What's produced in cardia? Mucus. What's produced in body and fundus? Acid and pepsinogen. What's produced by pyloric antrum, pyloric canal? Mostly. Well, mucus, of course. Mucus is everywhere, come on. But also gastrin. Because this is when food exits. Okay? Does that make sense? Know this difference, please. Okay, Gastrin secretion. How about we just merge a bunch of so we're gonna plug Gastrin secretion into the regulation of stomach activities, review them, and then we'll talk about ghrelin, somatostatin, you know, and serotonin and histamine. Okay? So what would increase Gastrin secretions if we talk about external stimuli? Smell of food or taste of food. Does that make sense? This is the cephalic phase. Are we clear? What will decrease Gastrin secretions? Being depressed, being bummed out, right? When you're not hungry. So again, that was cephalic. Now, internal stimuli. Okay, you said increased acidity. Okay, acidity or pH? Okay, increased pH. How would it change Gastrin secretion? Increase, of course. Does that make sense? What causes that increased acidity? Food. Sorry, increased pH. I'm sorry. My bad. So increased pH will be caused by food, right? Is there any gastrin secretion stimulated from the intestine? Remember short term? Intestinal gastrin. Remember intestinal gastrin. In the short term perspective, intestine will produce gastrin to a little bit increase digestive activities of the stomach. Okay. Now, what will decrease gastrin secretions in terms of the internal stimuli? A lot of fat where? In the duodenum. Basically, food in the duodenum, in the long-term perspective, will decrease gastrin secretion. Does that make sense? What should happen in the stomach to decrease gastrin secretion? Increased acidity or low pH. It's the same. Are we clear? So what does gastrin do to the stomach? What does it do to the stomach? Let's talk about stomach first. Gastrin stimulates release of histamine. Gastrin stimulates histamine, and histamine stimulates HCl. Are we cool about it? Okay? Now, what does it do to intestines? Gastrin. What does it do to the small intestine? How does gastrin affect small intestine? It increases peristalsis. What does it do to the ileocecal valve? Huh? Opens it up. What does it do to the large intestine? Mass movements. Yeah, also contractions. That make sense? Propels the foot down the road. We're good? Is that clear? So gastrin basically it is stimulated by food, and gastrin stimulates activities that will get rid of that food in the stomach. So which cells are directly stimulated by gastrin in the stomach? It's ECL, enterochromaffin-like cells. Ghrelin. What increases ghrelin secretion? Faster. Yeah, that's as far as you need to know. That's just physiological role is stimulation of the appetite. Whenever you are in the fasting state, in post-absorptive state, basically, ghrelin is going to be stimulated. Serotonin. What stimulates serotonin release? Food. What's the activity? Contraction of the smooth muscles. Histamine. Food. It is stimulated, well, indirectly, right? So it's produced by which cells, she said? Enteroendocrine ECL cells. Stimulates parietal cells to release hydrochloric acid. And yeah, that's kind of it. And yeah, gastrin stimulates histamine release. So say if you overdo Benadryl, what you get in addition to being sleepy? Constipation. Main stimulus for secretion of somatostatin. Surprisingly also food. But what are the effects of somatostatin? It inhibits digestive activities. It inhibits muscle contraction, smooth muscle contraction in both small intestine and stomach. It inhibits the pancreatic juice release. Okay. Parietal cells are stimulated, as we just mentioned, like over and over again. Histamine. Okay. That's histamine. They produce hydrochloric acid. Now, remember that hydrochloric acid production thing that we talked about? Okay, when water interacts with CO2, produces, which dissociates. Okay, sorry for handwriting. Okay, so you have to understand where CO2 comes from. Where? Blood. You have to understand hydrogen, where it goes. Lumen of intestine. Does it diffuse or is it pumped? Hydrogen is acid. What do you have in the lumen of intestine? More acid. It is pumped because it goes into the higher concentration. That makes sense? And bicarbonate, where does it go? No, this bicarbonate doesn't go to the intestine. It goes into the blood. Yeah, this one goes into the blood. That would be lovely, though. But it wouldn't actually, because if there's no food in the duodenum, then you don't need bicarbonate there. It's like having yourself a baking soda animal all the time. Mechanical digestion in the stomach, churning, you know. What you do have to understand is that when you have all that food in the stomach, there are small portions that go through the pyloric sphincter into the duodenum. Just let's be absolutely clear about it. Okay? We're good? Pyloric sphincter controls this movement. Now, remember we talked about effects of chyme in the duodenum on the gastric secretions and blah, blah, blah? Again, in the short term, it increases gastric secretion and empty, right? But in the long term, what happens? It decreases. In the long term, the effects of duodenal presence of food in duodenum, those effects decrease gastric secretion and gastric emptying. Okay. Cephalic gastric, so cephalic, just talked about it. Smell, taste. It works through the cortex and through the brainstem. Basically, all your majority of your digestive activities are mediated by brainstem. Okay. What inhibits depression? Mechanical stimuli increase gastric digestive activities. Distension. How do we call these reflexes? That's vagal. Well, vagus, because it's vagal. Integrative center for this reflex, medulla. Chemical stimuli. Chemical stimuli in the gastric phase that increase digestive activities. High pH, and just chemicals in the stomach. Which cells are targeted? Enteroendocrine cells. They produce gastrin. Which chemical change in the stomach inhibit? Low pH and basically absence of food. Which cells respond to these changes? G cells. Which type of nervous stimulation may inhibit gastric digestive activities? Which branch of autonomic nervous system will decrease digestion? S word. Sympathetic. Good. Chemical stimuli in the intestine stimulate gastric digestive activities. So remember, food in the intestine, intestinal gastrin, short-term activation. Got it? Stretch of the duodenum, which means food in the duodenum, leads to the reflex inhibition of stomach, and CCK mostly will also inhibit stomach activities. Remember, when chyme enters the duodenum, which two hormones are released? CCK and secretin. CCK mostly will inhibit stomach from delivering more food. We're good? Gastric ulcer. Anatomically, it's the hole in the wall. Maybe incomplete hole. lesion. What the main cause? H. pylori. What are effects of NSAIDs on the gastric secretion? Gastritis, they basically inhibit the mucus production. Is it a risk factor? I mean, gastric ulcer for something like cancer. Yeah, for stomach cancer. Okay, that's easy. Duodenum. Most important digestive organ. What empties in the duodenum? Liver? Pancreas? You said pancreas? Oh, stomach. Yeah, you're good. Stomach. Stomach empties in the duodenum. What else? Gallbladder, liver, and pancreas. Okay? Just imagine the anatomy. Where the secretions of pancreas and liver enter the duodenum. What's the name? Hepatopancreatic ampulla. Remember? Hepatopancreatic ampulla. Yeah, nerves control the duodenal activities. It's going to be the splanchnic nerves from sympathetic and vagus for parasympathetic. Nerves that control duodenum, are they somatic or autonomic? Autonomic. Blood vessels, superior mesenteric artery. What's important is that, remember, duodenum is connected to, well, the entire intestine is connected to the liver, especially small intestine, intrahepatic circulation. 3 structures that increase the surface layer. Anatomy. Identify them on the picture. Circular folds, villi, microvilli. Circular folds, they move food in a circular fashion, which keeps it in the duodenum for a longer period of time. Why is large surface area important? Absorption. Which type of epithelium? Simple columnar with microvilli on top of the cells. Does that make sense? Microvilli are membrane folds. What is in the lamina propria of duodenum? Pretty much I refer to this, mold. Mucosa-associated lymphoid tissue. Protection, okay? What's the function of lacteal? Absorption of fat and its lymphatic vessel, okay? Layer of microvilli on epithelial surface is the brush border. I'm not a Canadian. It's the. Importance of this layer. What is in the brush border? Yep, enzymes. Actually, in some digestive infections, what you can see is the flattening, destruction of the brush border, and then digestion severely impaired. Not so much digestion, but actually, no, digestion and absorption. Enzymes of the brush border. Carbohydrates are digested by lactase, maltase, sucrase, dextrinase. Proteins are digested by aminopeptidase and dipeptidase. No really lipid digestion involved, but there is nucleosidases and phosphatases that break down nucleotides. There's enterokinase. What is its function? What does enterokinase do? Activates? Huh? It activates something. Enterokinase, trypsin. Yep, trypsinogen to trypsin. Goblet cells release mucus. Enteroendocrine cells release CCK and secretin. Why is mucus alkaline? Because acid is coming from intestine. Paneth cells, what do? Phagocytose, yes. Brunner's gland, where is it located? Well, a junction, yeah. It's right here, the junction between duodenum and stomach. Why? Because it produces what? Alkaline thick mucus, which protects duodenum from acid secretions. Okay. Let me please skip this, okay? You can figure out from the name. Substrates for lactase, maltase, and so on and so forth. For enterokinase, just described aminopeptidase and dipeptidase. Well, peptides, nucleosidases and phosphatases, substrate are nucleotides. The benefit of having them in the brush border, you don't release them into the lumen, so they don't get digested in the lumen, like, because they're proteins, and they would be digested. Okay, secretin, produced by what? Enteroendocrine cells in the duodenum. What stimulates the production of secretin? Let's just go over the whole deal of regulation. So what is the stimulus for secretin? It's the result. Secretin stimulates the release of bicarbonate from pancreas. What is the stimulus? Just chem or something else. Something is in the chem. For secretin, huh? Acid. So acid is the stimulus, and it stimulates the production of bicarbonate. What effect does it have? So it's from pancreas. What effect does secretin have on liver? Bile production. Not so much, but to some extent. What about CCK? What stimulates CCK release? Huh? Fat in the chem. What does CCK do with pancreas? Stimulates the release of enzymes. Very good. And it also stimulates the contractions of the gallbladder. And here I want to make a plug for gallbladder. We talked about gallbladder as being, kind of, you can just cut it out. When gallbladder contracts, what is released in the duodenum? Bile. What does bile contain? Like, massive component of it is bile salts. Are they absorbed in the duodenum? Yes, they are. Look, gallbladder dumps bile in the duodenum. Bile salts are absorbed in the enterohepatic circulation. They go from duodenum to the liver, stimulate bile release, which contains bile salts. Bile goes to the duodenum, more bile salts, more absorption, self-stimulatory loop, sort of. Does that make sense? So, if you don't have gallbladder, what do you have to wait for? Bile being produced by liver in response to which hormone? Secretin. And it's rather slow. Does that make sense? Good? Are we clear about secretin? Are we clear about CCK? So I want to be very clear. Secretin, in terms of pancreas, secretin is bicarbonate. CCK, enzymes. We cool? Okay? Can we move on? This is what we just did. CCK in response to acid, bicarbonate production. Secretin in response to acid, bicarbonate production. CCK in response to fats, enzyme production. Okay? Mechanical digestion in the intestine. Segmentation and mixing. How long? Okay, if the question implies numbers, please don't memorize the numbers. I beg you. Okay, there's going to be. I don't think I ask how many ATPs are ATPs and ADH, have an idea about those numbers. Yeah, that's probably the only numbers you will have to memorize in potentially the entire course. Okay, and maybe volume, not volume of urine now. Liver, anatomically, what I will ask you, I think I have it on the anatomy study guide, but what I will ask you, like 4 lobes mostly, and a couple of ligaments. Covered with connective tissue attached to the diaphragm. Elementary building blocks. So when we talk about liver lobules, understand, we've got plates of hepatocytes, right, with sinusoids running between them. Sinusoids carry blood. Sinusoids are capillaries. They carry blood. They're very permeable. Blood in the sinusoids comes from portal arteries and surprisingly veins. Okay? This blood mixes and goes to the central vein in the center of the lobule, which then those veins, they bigger, eventually get into the hepatic vein gets into the vena cava. And where does the blood from liver go? To the heart, and from the heart it's delivered all over the place. Okay? Keep that very, this whole blood pathway, keep it in mind pretty well. What is the function of bile canaliculi? Come on. Bile canaliculi. They are vessels that carry bile, people. Okay? So bile goes to the bile ducts. Okay? Bile ducts then also they kind of join together like river, okay? Like rivers join together. And they all come down from the liver in the form of one common hepatic duct. Common hepatic duct merges cystic duct, forming bile duct or common bile duct, and then add the hepatopancreatic ampulla, bile duct merges with pancreatic duct. That's the pathway that I want you to know. I was pretty explicit. Which cells produce bile? In the liver. Come on. Hepatocytes. You can speak up. I can barely hear you. Okay. Bile goes into the duodenum. Okay. So, components. Know the function. Like bicarbonate. What does it do? Neutralizes acid. Lecithin and bile salts. What do? Emulsify the fat. Why do we need the fat to be emulsified? Say again? If you would go into the, what is the word? Sewage system in New York. You've heard about the clumps of fat that were discovered? No? Well, that basically, and they were discovered of all places under the Chinatown. Restaurants, Chinese restaurants, mostly Chinese. Not that I'm chauvinistic, but they dump enormous amount of fat directly into the sewage. And when it cools down, it solidifies. There are, by clump, I mean, they're like the size of a car. So we're talking like tons. Like imagine that several tons of fat, which also traps everything else that goes into the sewage. So the aroma is perhaps unbelievable. And it's a problem because then people have to go and clean it, which is a wonderful job by itself. So bile can travel. Fat can be in a water. The problem is that it's going to travel as the one gigantic globule, right? So you need to break it down to emulsify, to increase the surface area, so fat can be digested. Cool? Bilirubin, where does it come from? It's a product of degradation of red blood cells, heme, component of red blood cells. So bilirubin has to be excreted in bile because it is attached to the albumin in the blood and cannot be excreted by liver. Jessica, to your question, I double checked it. Hepatocytes manage to separate bilirubin from albumin, and they leave albumin in the bloodstream. Remember you asked about the filtration? I double checked. So bilirubin goes into the bile, hepatocytes kind of pull it out, dump it in the bile. In the intestine, what happens to it? There are two options for bilirubin. It can be excreted with feces, or it can be converted to urobilinogen, which then enters intrahepatic circulation and gets in the blood and filtered out from the blood by the kidneys. Okay? Bile salts are amphiphilic, both hydrophobic, hydrophilic. What do they do to lipids? They emulsify them. How do we call these assemblies of bile salts and lipids? Micelles. Okay. Kidneys cannot excrete bilirubin. We just talked about it. Bilirubin is bound to albumin. What happens to it in the intestine? Bilirubin is converted into the urobilinogen. Urobilinogen gets into the blood. Urobilinogen may still, look, urobilinogen may still stay in feces. Okay? Does that make sense? But it will also get into the bloodstream. Liver in the detoxification, what the? Okay, what I mean by that? Just know that liver is the main metabolic organ, okay? So Kupffer cells are the phagocytes, macrophages that will kill the invaders. And in terms of the metabolism, first of all, it digests most of the toxins, such as ethanol, for instance. What does it do to carbohydrates, liver? It converts all other sugars like galactose to glucose. It can bypass certain steps in glycolysis and convert fructose into the fat pretty much directly. It pretty much stores carbohydrates in the form of glycogen or converts them to fat. What does it do to fats, liver? Remember? HDL, LDL? What does liver produce and ships out to the tissues? HDL or LDL? From liver to tissues is from liver to tissues, it's LDL and VLDL. From tissues back to liver, it's HDL. Liver makes proteins, right? A lot of proteins, albumin, coagulation factors, a lot of them. So albumin, coagulation factors. Intrahepatic circulation, just discussed. For bile salts, it's important for cholesterol excretion. How do bile salts regulate their own level by intrahepatic circulation? They get absorbed in the intestine, go to liver, stimulate their own production. What happens to the excess bile? Gets into the gallbladder. What happens to the bile there? And concentrated, stored and concentrated. There's no submucosa in gallbladder. What is the pathway of bile release? We just talked about it. I'm going to ask. Mechanism of gallstones formation. Bile gets concentrated cholesterol and calcium form gallstones. What causes pain in case of cholecystitis? Blocks the duct. Treatment options: get rid of the gallbladder. Risk factors: lack of motion, just move around. And, being old also doesn't help. Pancreas, anatomically, be able to identify, know that acini produce pancreatic juice, anatomical parts, head, tail, body, secretes juice. Chemical component of the pancreatic juice, it's bicarbonate. Does that make sense? Remember we talked about bicarbonate synthesis. This one. Okay, bicarbonate gets into the ducts in the pancreatic juice and goes into the lumen of intestine. And where do protons go? Hydrogen ions? Blood. Enzymatic components. Know the activation cascade for trypsin. And then the trypsin activates chymotrypsin, elastase, carboxypeptidase, and phospholipase. Know the substrates, like protein, lipids and phospholipids, cholesterol, splits cholesterol from the proteins. And this is DNA and RNA. Well, RNA and DNA. Okay. Quickly review absorption, digestion, and absorption. Proteins, where are they digested? Come on. Stomach and intestine. Lipids, mouth, stomach, intestine. Carbs, mouth and intestine. When proteins are digested, what are they digested to eventually? Proteins. Amino acids. Amino acids, how are they getting into the epithelial cells? Well, amino acids are already, that's just basic stuff. That's fundamental, right? How do they get? What? No. If it's not diffusion, then how do they get there? Active transport. Secondary active transport. They co-transported with sodium. Sodium diffuses, and amino acids get the energy of that diffusion. Why sodium diffuses in? Because the concentration of sodium in the epithelial cells is low. Why is it low? Sodium-potassium pump at the basal side. So basal apical, that's sodium-potassium pump, right? Okay? Does that make sense? It keeps it low. Carbs, what are they digested to? Monosaccharides. Monosaccharides, how do they get to epithelial cells? Secondary active transport. And then what happens to them later? They diffuse into the blood, as well as amino acids. Facilitated diffusion. Does that make sense? Triglycerides. What happens to them? What happens to triglycerides in the micelles? They're broken down to what? Fatty acids and monoglyceride. Okay, not completely broken down. So they enter as the part of micelle. They enter the epithelial layer. What happens to fatty acids and monoglycerides in the epithelial cells? They recombine into triglycerides, and triglycerides get into the lymphatic system, lacteal. Are they going there by themselves, or are they packaged in something? Chylomicron. microns. Remember, triglycerides are packaged with the proteins. Okay. So we got this. Okay, is that clear? In which parts carbohydrate is digested? This. What? Well, I'm asking, is that clear? We just reviewed it. So I want to make sure that this is all good. Nucleic acids digested in the intestine. Okay. Do nucleic acid have important nutritional value? No. Large intestine anatomically. Cecum, appendix. Ascending, transverse, descending sigmoid colons, rectum, right? Absorptive, does that carry out a lot of absorption? Large intestine. No, not a lot. Mostly water and just residues. Residual water, I mean. Cecum, what does it store? Bacteria, as well as appendix. Commensal bacteria, well, they help us to digest, really, and immune functions and all that kind of stuff. Obesity, function of rectum. What's the function of rectum? Expulsion of feces, right? Anal canal, anatomical lining is stratified squamous. Anal columns and sinuses produce mucus for the lubrication. Voluntarily, yeah. So columns and sphincters know them, and external, internal sphincters know them. Anatomically know. Which one is under voluntary control? External. During the defecation, what is the reflex? Stretch of rectum. Signal goes into the spinal cord. Spinal cord produces motor response and relaxes. Internal sphincter. And external is relaxed when we let it to relax. Basically, what we do, we consciously inhibit sympathetic system when we relax external sphincter. We inhibit sympathetic, increase parasympathetic stimulation, it relaxes, boom. Amenta, okay? So, question? Greater omentum, lesser omentum. Those little structures, know them. Okay, mesentery. Now, epithelium, simple columnar, absorptive function, water. Intestinal glands secrete mucus, goblet cells. Mucosa-associated lymphoid tissue, immune responses. Role for Tinea coli, what do? Tinea coli, they help mass movements, right? Haustra are the bags. Yeah, Tinea coli and Haustra. Identify them on the bigger picture for the large intestine, okay? Mass movements. What's the stimulus for mass movement? What can be stimulus? There's like one 4-letter F word. It's appropriate food, yes. Food, really. You eat, eventually gastrin increases and stimulates mass movements. Smooth muscle cells in the colon. So slow waves are pretty much pacemaker-mediated waves of slow depolarization. Okay? And if there's an additional stimulus like acetylcholine or stretch, it causes contraction via initiation of action potential. Now, when food is moved down through the, it should be SMC, smooth muscle cells. When food is moved through the intestine, what happens to the downstream part of the intestine? Is that relaxed or contracted? Downstream. When you, relaxed. Upstream, contracted. So what stimulates the contraction? No, I'm talking about neurotransmitters. ACH or substance P. Remember, you have a stretch, okay? Stretch is mechanical input, sensory input. That stretch initiates two reflex arches. One arch goes upstream of food. That's where the contraction happens. Two excitatory neurotransmitters, ACH and substance P. If you skipped on neurotransmitters in AP1, now it's time to lament about it. So ACH and substance P are excitatory neurotransmitters. They stimulate the contraction. Downstream, have two inhibitory neurotransmitters, NO and vasoactive intestinal peptide, VIP. Good? Okay, we got the defecation. What's the difference in this reflex between infants and adults? Infants cannot control it. Hopefully adults can. Vomiting. What can cause vomiting? Irritation, right? So chemical, or obstruction. So basically, we have two stimuli. Again, chemical stimulus, like you drink a lot of alcohol. It's not necessarily overstretching, you know. You're not going to get overstretching if you drink a bottle of vodka. But it's going to be a lot of stimulation, chemical stimulation. You will vomit. Obstruction is the mechanical. You block, food accumulates, you have a stimulation, you vomit. So that sensory input from the stomach reaches vomiting center and medulla. And medulla, essentially, what does it induce? How would you call it? Inverse peristalsis, yes. Okay? Part of sinus medulla. Electrolytes are absorbed mostly in the, actually, water and electrolytes absorbed in duodenum. Okay? There is absorption in the large intestine. That's residual. What I want you to know about the absorption of electrolytes. Few things. Number one, sodium. How is it absorbed? Active transport at basal side, diffusion at the apical. How is water absorbed? Osmosis, it follows sodium. How is potassium absorbed? After water absorption, potassium concentration increases and it diffuses into the epithelium. What regulates calcium absorption? Indirectly, huh? Which hormone? PTH. Indirectly via stimulation of vitamin D, right? Is it absorbed like constantly or only when needed? Only when needed. What regulates iron absorption? Well, when you need it. When you need iron, you absorb it. We clear? What's that? Yeah. Celiac disease, which component of plant-based food causes it? Gluten. Basic mechanism, gluten forms immune complexes, and your immune system attacks those immune complexes. Are we good? Okay.